Hey everybody, and welcome to the first part of Unit 6, Lesson 2. So what we're going to be talking about here is protein synthesis, and anytime we talk about synthesis, that means to make. So we're talking about making proteins, and what you're going to find out is proteins are going to serve a very vital and major part of our structure, function, everything that we're going to do. So let's settle in here and we'll kind of talk about protein synthesis and why it's important and what is a protein also. So the first term is this central dogma. And really when we talk about the central dogma, we're talking about the process and the flow of going from your DNA to then making what we call mRNA and then what we call the protein. So the central dogma is just that flow of where we're going to go from one to another to another. So Crick proposed that in the first step of the process, the information flowed from DNA to an intermediate molecule of RNA, which we are going to call mRNA, which is going to be our messenger. And then that mRNA is then going to go to a ribosome, and then it's going to go and make a protein. So as we scroll down here, it says each protein is coded for by a section of DNA called a gene. So a gene is a piece of DNA. It's found in our chromosomes, and every single gene is found in the same spot on each individual organism. So for example, if you're looking at a gene for the type of ears you would have, that gene is going to be found on a specific chromosome in humans, and in every single human, you're going to find the gene for ear production on the same chromosome. So it's the same for everybody. Here it says humans have around 19,000 protein coding genes, meaning that there are 19,000 locations in your DNA that are going to code to make a specific protein. But it also says that doesn't necessarily mean how complex the organism is. And it says, for example, a grape, right? A grape plant is going to have over 30,000 genes. However, a grape is much more simple of an organism than a human. So there's no correlation between the number of proteins versus how complex they are. Okay, so what this graphic is showing is here's a chromosome, and here is it's broken down, and now it's into its simplest form is a double helix, and it's saying that the gene for any given protein would be like maybe from here to here. Okay, and then maybe this one would be a different gene for a different protein. And right here would be a different gene for a different protein. So a gene is just a section, okay? As we scroll down here, here's just a chart that talks about all of the different functions that proteins might have. So they're going to be storage, where we're going to store protein, and this is albumin, is what we call where it's found in egg whites, right? So egg whites is a storage for protein for that baby chicken that's going to be born. Those proteins are going to start and help provide their structure. Transport. So globin, this is a protein that's found in red blood cells that's going to help carry oxygen throughout the cells. You may have heard of hemoglobin, right? So maintain a homeostasis. Proteins are hormones. Proteins are antibodies, right? Right now we've got COVID-19 and everybody's trying to build up antibodies through the vaccine to where we can help our immune system if we were to get COVID, okay? So those are proteins that we're using. Movement. So here are two different types of proteins that are going to help with our movement. This one's going to be found in muscles, okay? And this one's going to help move things and other proteins and transport things inside the cell. Obviously, protein is going to be structure. It's going to have our cytoskeleton and muscle fibers, but it's also going to be the structure that's going to be found in hair and nails, and also in other organisms like feathers and horns. Protein is going to be used to make hair and nails in humans. Catalyzing reactions. So enzymes are catalysts. They make reactions go faster. So these are other examples. And enzymes we've talked about when we talked about replication, they all end in ASE. So here are two types of enzymes that are going to help break down sugar. So maltose is a sugar, and maltase is the enzyme that's going to break it down. Lactose is the sugar that we find in milk, and lactase is the enzyme that's going to help break it down and get it into a sugar we can use like glucose. Okay, so as we scroll down here, this is kind of building a protein. You can notice that all these individual colors are different types of bends and turns. What you're going to find out is the shape is what's very important to a protein. If the protein changes its shape, then it changes its function and it won't work. And we'll talk about that later when we talk about how we denature proteins and we change them. Okay? Okay, so now let's talk about the steps in protein synthesis, how we're going to make proteins. So there's going to be two processes. The first one is going to be called transcription, and the second one is going to be called translation. Now remember when we talked about the central dogma, we said we were going to go from DNA to messenger RNA or mRNA. That first part is going to be called transcription. So it's the process of turning DNA into this mRNA or getting the code. Okay, so what we've got right here is we're going to go from DNA to mRNA, and that is going to be transcription. Okay, transcription. Now the second part is translation, and translation is where that mRNA is then going to be converted into a polypeptide, which is a protein. Okay, so as we scroll down here, let me close that out. As we scroll down here, this mRNA is going to leave the nucleus. So here we're in the nucleus. This mRNA is going to leave the nucleus and it's going to get out to a ribosome. And out of a ribosome, we're going to go from an mRNA and we're going to produce a protein. And that's called translation. So there are the two parts, transcription and translation. Transcription takes place in the nucleus, and we're going to go from DNA to RNA. And when we get in translation, we're going to go from mRNA to a protein. Okay, as we scroll down, the last thing we're going to talk about is comparing DNA and RNA. There are a few differences. Okay, so here we've got DNA, and you can notice that we're going to have deoxyribonucleic acid, and in RNA we're going to have ribonucleic acid, and that's because the sugar is different. Okay, so then we're going to have a ribose sugar versus a deoxyribose sugar, and it talks about it right here. Sugar phosphate backbone. Okay, and RNA is going to have a ribose sugar backbone. Okay, now also you can see another difference is that DNA is double-stranded, right? You can see the two strands, and RNA is single-stranded as it works through. Now, the last thing is that you're going to see cytosine and cytosine. It's found in both of them. That is not different. Guanine is also found in both of them. That is not different. Adenine is found in both of them. That is not different. However, you'll find right here that we have thymine in DNA, but we do not have thymine in RNA. We have another, it's uracil. And it says right here it's going to replace thymine. So instead of C, G, A, and T, we're going to have C, G, A, and U. And A and U are going to go opposite one another. So as we look at here, let's go ahead and fill out this Venn diagram together, talking about whether they're found in one or both. So as we scroll up here, sugar is deoxyribose. Well, we know that that is going to be found in DNA because that's what the D stands for. Uracil is only going to be found in RNA. Nucleic acids, both of these are types of nucleic acids because they're going to have a phosphate, a sugar, and a base. A sugar and phosphate backbone, that's going to be both of them, okay? Sugar is ribose. We know that that's going to be found in RNA, okay? Single-stranded. Now, remember we said that RNA is going to be single-stranded. Whoa, wrong side. And that's actually going to play a big reason on why we need that RNA. Thymine. You're going to see that thymine is going to be in DNA only because we have uracil and RNA. And then the last one, double-stranded, that's going to be our DNA because we know that RNA is single-stranded. There's thymine and uracil. We're going to have deoxyribose and ribose, and these are the ones that are both. Okay? That is going to be the end of this exploration. Hopefully you were able to learn some things about proteins. And then as we get into the next one, we'll talk about transcription and translation.